Afternoon, everyone. I feel nervous because of studio camera. My name is Roza. I'm a research student in this laboratory, and now I want to tell about my experiments. This experiment consists of some observation. I have done 3 observations that I want to share with you. Firstly, I want to tell about my outline, this objective, observation 1, 2, and 3 that I want to share with you. Observation 1 use UV camera, and observation 3 use UV visible spectroscopies. There are some relationship that I want to tell with you next. Observation 2 I use double beam spectroscopies. That's to investigate juice acidity contained in lemon juice, and conclusion activity. This is objective study non-destructive measurement acidity content and orange in UV region with spatial illumination. Actually, I am focused to use spectroscopies, and our laboratory provide UV visible spectroscopies that I will use to next my experiment. Those my observation. I use UV camera, xenon light source, halogen lamp that's has power 150 watts, double beam spectroscopies, and the last one UV visible spectroscopies. This I will introduce the first light source, the light Q. There have spectrum intensity like this. That means I can use for UV region, because there are spectrum like this. And actually this spectrum can use from UV region to NIR region. This is my first observation. Use UV camera because I think if UV camera can get capture in that means spectroscopies also can get spectrum intensity in UV region. So that's why I need comparison between UV camera and UV visible region. This is the result. Based on this result, I wanted to measure UV by UV visible region, UV visible spectroscopies. This is my observation. Second observation use double beam spectroscopies. This is some iPhone paper that this paper talk about UV double beam spectroscopies, and this is my second observation. I use water, vitamin C. That's very thank you for you, Jasang, give me sample to my experiments, and lemon juice. So I compare those sample, and based on this. Based on this spectrum, I got conclusion like this, and also this point supported by some literature. The water, there is no high intensity in from UV to visible, and because of contained, I think because of contain acidity or other, there are some spectrum, even though they are saturated. in UV visible in UV region. So to overcome this saturate, I think it's better to add NaOH and measure again to use. Measure again to use by spectroscopists. So this is my third observation. This use halogen lamp. Especially I use UV visible spectroscopies provided by our laboratory. I measure non-destructive without make use. I try to use 3 halogen lamp and detector by UV visible region. In the field like this, I take the picture. This condition of my third observation, but this is the problem because of maybe an higher region, the temperature very high and make juice become burn. And I try to use this filter to avoid the high temperature, but because of there is no cannot cut because there is no high transmittant in UV visible region, so that's why the filter cannot work full. Like this burned by temperature of lighting. And this is the result. There is no result. That's my problem. And, even though you use 3 light source, and there is no intensity I won't share with you. This. Also, then I want to use the filter like this to avoid UV. To avoid the high temperature, the spectrum of filter like this. Conclusion: need just light power to get intensity spectrum of orange. The second one, possible to obtain transmittance mode in UV region by adjust power light with object. And the next plan is increase several the light source with several illumination and use filter to get spectrum in UV visible region. Thank you.
This one.
So what kind of results are you expecting?
Result?
Yeah.
To get transmittant in UV region. I hope I can get transmittant in UV region by some several illumination.
So
adjust.
if in an ideal condition, can you, if you have the ideal condition, can you draw the line? What are you expecting? The line is go, how the line go?
Oh, I hope the line. Sorry, like this, round. Yes, round.
A peak.
Huh?
A peak goes up.
Up,
Yeah.
up in UV region. Like this. That's my expectation.
Also, go back to the slide compared with water, vitamin C and lemon juice.
Ah.
No, yeah, that one.
This?
Yeah. In case of lemon juice, the shape of graph is not consistent with, not same.
Juice.
Yeah. Is this due to the
Yes.
concentration of juice?
Yes, there in the visible region, I think this depend on color. The color of juice, I think many kind color of juice. So that's why several various in the UV spectrum in the UV in the visible region because of color, I think.
Any other comment? Question? Did you talk about these materials? How did you get the lemon juice? How did you get the vitamin C? What is
Yes.
vitamin C?
This I tried to use Yulia Sang's sample. There are
Where did you describe about that? How to get the vitamin C? Vitamin C, just vitamin C is a vitamin. Is that juice or
liquid.
liquid?
Liquid. The color
Yes,
same.
then where in the material you explain about those? Material section.
Yes.
Lemon juice, how many type of, how many?
A sample.
Yeah.
The sample.
5 samples.
19 sample.
19 samples. But
Yes.
for 5 samples. Yeah, we cannot follow your presentation. How did you get the lemon juice?
Lemon juice I extract from lemon.
Extra?
Yes, extract.
Extra.
Made
Squeeze.
Squeeze. Yes. And,
When did you explain about that?
1 month.
1 month? No. Today's presentation.
Today's presentation.
Show us that lemon juice material. What's the difference? Those 5 juice?
Concentration.
The concentration is where? Where is the information? The concentration.
Yes, maybe it's I.
You didn't do
Yes.
in the presentation you have to make clear about the material, method, and result.
Yes.
Also on the ground, I always tell you, unit, no unit, and why absorbance is a minus. Then no description on these. This is sample.
Yes.
Sample. Sample
one.
1 here, sample 2 here. Why is this not organized? Why this is 1, 2, 3? I'm not sure.
Yes.
This is 6 samples here.
Yes, 6 sample. That's
Overlapped all of them. But we don't know the difference among them. This is absorbance.
yes, absorbance.
Usually absorb, you'd better do right absorb. Then, yeah, of course, this nanometer. Nanometer?
Nanometer.
Nanometer.
Yes.
Okay, would you show us from the top? Maybe I couldn't understand. I tried. For example, this one, or anyone. We can't read this. Can you read this? What did you tell about this? What is? Can you read? Then what is?
It is the mechanism of.
So what do you want to tell us about this from this? Maybe no one can understand this kind of flow, not block diagram.
Yes.
Not easy to understand. And before this, what is? What is? I can't see anything. What did you want to do? Maybe it's difficult to follow your presentation. Yeah, okay. So you'd better discuss with your group. Maybe group leader is who is a group leader? Sequin is a group leader. Please suggest him to improve. Yeah, this is no good. It's better not to open this presentation to the world. Maybe. Okay. Yeah. Anyway, so improve more.
Thank you very much.
Anything else? Okay, maybe too much. Okay. Okay. Thank you very much.
Thank you very much.